from the campus of the University of Nebraska at Omaha. Consider This, a program of issues and information behind today's news.
In the global race to find a vaccine, Oxford University just jumped way ahead of the pack. Human testing is already underway, and scientists say they're hopeful it will be available by September.
Well, personally, I have a high degree of confidence about this vaccine because it's technology that I've used before.
Technology they had already developed in previous work on inoculations for other viruses, including a close relative of COVID-19, giving the lab a step ahead. The vaccine takes the coronavirus's genetic material and injects it into a common cold virus that's been neutralized so it can't spread in people. The modified virus will mimic COVID-19, triggering the immune system to fight off the imposter and providing protection against the real thing.
Hi everyone, I'm Kathy Wyatt. Welcome to the 7th edition of a special weekly series on Consider This, COVID-19. Today's focus, race to a vaccine. Joining us via Zoom to talk more about it is Dr. David Brett Major. Dr. Brett Major is a professor in the Department of Epidemiology in the College of Public Health at the University of Nebraska Medical Center. Doctor, welcome.
Thank you. Thank you for having me, Kathy.
Let's start by having you walk us through examples, right, of a couple of vaccines developed to prevent diseases, maybe back in the day and then a few more recently.
The vaccine history is really fascinating, and it's ancient. With smallpox in areas of the Middle East, they used to take wounds from smallpox and inoculate it in non-obvious parts of the body to try and get a response to the smallpox so that later in epidemics, you would not get scarred. And that was a practice over many, many years. And then an advocate against smallpox, the wife of a diplomat, Lady Montague, went out to the Near East and lived there, saw the practice, got very interested in it, and brought it back to England in the early 18th century. And that started the practice of inoculation, of variolation. Variola is the other name for the smallpox virus. And that went on for many years, and people realized that that cut down how many people died by about 1 in 10. You could still die that way, but far fewer were affected. And so then, late in the 1700s, an English scientist and physician named Edward Jenner, who had grown up on a farm and remembered dairymaids commenting that they were protected from smallpox because of cowpox. It was farmers who identified vaccines, right, relevant for Nebraska. And so he started using vaccinia virus, cowpox virus, in order to protect people. And that was the first vaccination. And then almost 100 years later, Louis Pasteur, a chemist, got interested in this whole issue of getting people protected. And he started work in chickens, and he took a bacteria for cholera in chickens, and he passaged it over and over again, and it got weak. And then he gave it to chickens, and it protected them. That was the first attenuated vaccine. He did work with rabies, where he inactivated rabies, which brought him a lot of fame. And so he really created both attenuated and inactivated vaccines. Then we had a bit of a pause, and you have to get really all the way up into the mid-1900s in the work of Sabin and Salk and polio to really see the rise of vaccinology, of working in vaccines. The vaccines that we use most commonly in society came quickly, in about 20, 25 years in that window, where we started to see similar approaches for measles, mumps, rubella, tetanus, and eventually against hepatitis B. It has become more and more elusive to get good, effective vaccines against the other threats. Part of that is because we realize that vaccines have to be generated deliberately and safely. Part of that because we start tackling harder and harder threats. So the HIV virus is a perfect example of that. We've had really fantastic vaccinologists working on that for decades. and it's been slow going. And it was actually in that work that I started getting involved with using adenoviral vectors. And so it's actually common in that space to do it. So I was interested in the Oxford video that you just showed, and I'm sure we'll get back to that. Vaccines, more recently, the emergency use vaccine for Ebola virus that was put onto a vesicular stomatitis virus, basically a plant virus. And so it was interesting to see technology start to change, where more and more people started looking at other kinds of viruses to use as a way to deliver things that would spark the immune system.
Okay. This might be a good time in the interview, I know it's early on, but to ask you a little bit about your background. How did you become interested and get involved in this realm?
In vaccines in general,
yeah.
I was training in internal medicine, and my mentor, my attending physician was Nelson Michael, who's prominent in the HIV vaccine development space. And he really got me interested in the idea of working against HIV, which I found really fascinating. And he had me working in his lab with Mary Maravich, who's currently with the Division of AIDS at NIH. And I got to work with John Mascola, who's head of the Vaccine Research Center. So some really fantastic people. I'm one of a whole generation, really, of clinical trialists and functional immunologists that work with those people. And it's just the interest that your teachers and mentors have in science and public health, and watching discovery come together in that way.
So that is what keeps you most engaged, which was my next question, right? This is a pretty complicated topic, and so there has to be some component that really draws you in on a continual basis, right?
Vaccine work can be very frustrating work, and so it has to be something that continues to draw you in small steps, and continues to draw you in on a long horizon. It's not principally what I do now, but I've really appreciated opportunities to work with those folks who've been able to keep focus on developing vaccines over long horizons and platforms for vaccines.
Okay, what would you consider, Doctor, to be the most significant infectious diseases?
That is a very hard question, because it gets to an idea about how we weight and think about significance. Right? Globally, HIV, malaria, and tuberculosis remain pervasive threats. 24,000 people a week die of HIV disease. Tuberculosis numbers are mixed with that. And so when looking at global burden, it's hard to compete with that. Mm-hmm. Children under the age of 5 suffer very dramatically from acute respiratory illness, including influenza and other respiratory viruses. Mm-hmm. And so they're a significant cause of death in children. Viruses and other diseases of the gut are also important in that age group, mostly bacteria, but viruses play a part. Polio can be resurgent, and then you start getting into this group of diseases that do not necessarily have a lot of regular impact in communities, but have the potential to be very, very disruptive. And of course, we skipped over diseases like dengue and other ones that have hundreds of thousands of people affected, in cholera. Mm. But we have these emerging diseases where you get disruption of stable systems. And I was on a priority panel that used to have this conversation at WHO, and it came up with a priority list. That's actually the list where people started talking about disease X. And these diseases are ones that emerge and either are so communicable, so dangerous to individuals, or there's something about the way that they move from people to people, that they disrupt the way that we live, that we consider them high consequence. So that's the high consequence pathogen space. On that priority list, we had included SARS, severe acute respiratory syndrome coronavirus, MERS, Middle East respiratory syndrome coronavirus, and other novel emerging coronaviruses. which we are now experiencing. So we get points for stating the obvious a few years ago, but we were one of many.
Well, we want to put up a graphic for our viewers to kind of illustrate what has transpired in terms of developing vaccines over the years. So let's put that up. And as we do that, I mean, if you look at it, it appears that the time it takes to develop a vaccine has been reduced pretty dramatically. I mean, if you look at the top 28 years, all the way down to the possibility of just 18 months for COVID-19. What are you seeing on that slide, Doctor, that's important to note?
Well, the numbers are dropping, and the green box there is very aspirational. I think this post-tetanus, measles, mumps, rubella, polio vaccine era is a really interesting era, and our thresholds for feeling that the importance of having a vaccine and preventing certain diseases go down. We get more and more interested in interrupting transmission of diseases that we took for granted in many ways before, and inappropriately so, because our vision was obscured by diseases that had even higher impacts. And so I think that strikes me. But certainly where you're highlighting, and I think appropriately, the interest in getting vaccines out quickly is just rising.
Yeah.
I think 18 months is a reasonable estimate now. The candidates for Ebola virus disease actually emerged pretty quickly during that epidemic. The challenge really became getting appropriate efficacy data, get how it works in people who are actually experiencing the event, and that takes time. And you have on this slide rotavirus, and rotavirus is a great example of an important vaccine that was important to field, but had a problem. And the approach to rotavirus vaccine had to be changed. And so highlighting the importance of doing these things deliberately.
Yeah.
But you can do deliberate things quickly. And so that's always a balance.
Before we go in further to COVID-19, why are there some vaccines for some diseases and not others, like coronavirus?
Yeah, well, I think the question about coronavirus is mostly that we don't see a lot of it, and it doesn't hang around a long time and infect lots and lots of people. I'm not yet convinced that coronavirus is difficult to tackle immunologically. I think that vaccines that protect you from what happens in the lung are a challenge, and we can talk a little bit about that later. But coronavirus itself has a sticky bit. It has a sticky knob. When you get on the internet and you search on coronavirus and look at a picture, you see a crown, and it is it looks like a crown because it has something called a spike protein that looks like a knob, and those knobs are all around the surface and pretty predictably. And so I think our body sees parts of this virus relatively easily, and I think we'll learn how to target what part of those sticky bits on the virus are best to trigger our immune system to take the kind of action we want it to take. So I'm not sure that coronavirus vaccines are going to be particularly hard. There was, in the grand scheme of things, versus something like HIV, where the virus is very mutable. It's very easy for the virus to change. Individuals can actually be infected with many variants of the virus at once. They almost swim in a soup of differences when they travel around, and they embed into cells. It's a retrovirus. It retrogrades. It goes into the genome of the cells, and so to get rid of it, you have to get it out of the cells or keep it from getting activated from that DNA. So there are viruses that are harder, but this issue of body compartment is an interesting one. We saw this a bit in disease and Ebola, and I keep going back to Ebola just because it was our other recent large emergency that people think about. But in Ebola disease, the virus would hide in certain parts of the body, and they're called immunoprivileged sites. And so if your body does not usually put immune protection in places, that's a place for the virus to hide. And viruses are small, stupid, and everywhere. They are very good at getting wherever they want in the body. And so that can be a challenge with some viruses. The lung is a little bit different. It's not immunoprivileged exactly, but it is different. So years ago, people worked on a vaccine against plague as part of our effort to protect against bioterrorism. And it was hard to demonstrate that the immune response would happen in the lung in the way that you would think to protect. And so it's not always straightforward how that happens. Occasionally, in the case of dengue, it's a problem of lots of different kinds of dengue being around, and the viruses being just different enough that getting the immune system to be able to target all of them rather than just one or two is a problem, and that's also an issue in HIV.
Okay.
For influenza, the problem's been different. It's that the proteins that are on the flu, one, they change a lot, and there are lots of different influenza viruses. So we would have this problem if there were lots and lots of coronaviruses that we cared about too. But part of it is that the proteins go through a lot of change, and they have a lot of sugars and other things that make it a little bit harder to develop an immune response that's really active and durable over time. And that's why flu vaccines, while very, very important for overall public health management of influenza, and why it's still possible to have illness once vaccinated.
When it comes to COVID-19, we saw a video at the beginning of the program, Oxford University in the UK. Who is making strides in coming up with that vaccine, doctor, in addition to them?
So you gave me homework when you asked to have this conversation, and I spent a little time looking again at World Health Organization, Coalition for Epidemic Vaccines, CEPI, CEPI, and the literature. And it's interesting looking at them. And by the way, anyone interested can go to www.clinicaltrials.gov and actually search on a disease and a vaccine and see all the different trials that get registered around the world. So plenty of ones that are happening in China and are not necessarily using US technology or regulations still list themselves there. So it's good for somebody who's particularly interested in this. There are, to my knowledge, 6 vaccines that have entered clinical trials. The Oxford one that you mentioned is from a group that worked with chimpanzee adenovirus. It was a chimp ad3 platform that they used actually for their Ebola work, and that vaccine is still in development. The use of adenovirus as a vector, as a way to bring material into the body has been going on for a while. I worked for the great Merlin Robb, who's a clinical trialist guru, and he helped with mixes of pox viruses and adenoviral vectors. And so pox viruses, from smallpox, but that same vaccinia, for those of us who were vaccinated for smallpox, either because we're old enough or we were in the military or something, that same virus is very big, and you can put lots of inserts into it. So there's a pox virus version, there's an adenovirus version, there's a couple of versions where they tried to modify immune cells. There's a version where they've made a couple of proteins and they just put the protein into the body. It's interesting that none of the main ones listed by the major funders are ones that have actually are approaches that have actually resulted in fully licensed vaccines, but they do represent the breadth of technologies that have come to be understood as the next way to approach it.
Okay. Well, and finding and approving the vaccine will be one thing, but making sure there's enough of it to go around is another, right? So talk about just that piece of it.
That's a fantastic point. And our experience in this is recent. So when H1N1 was a problem from 2009 and 10 especially, but there were also subsequent waves, that vaccine used the same infrastructure as seasonal influenza. And seasonal influenza remains a problem for people in communities. And so those manufacturing resources had to be diverted in order to produce vaccine. There was a point about a third or a little bit more in the epidemic where the group of individuals who make the recommendation for how an annual vaccine for influenza, for flu, ought to look, they met in WHO and they said, all right, it's time to stop all of our production for seasonal flu. And you know how many people get seasonal flu vaccine. It's time to stop and switch all of that manufacturing capability to be able to produce and distribute an H1N1 flu vaccine. And they did that. They were fortunate that they did it at a time when they were essentially trailing production anyway. They were near the end of their production for influenza vaccine for the season. Mm-hmm. And they weren't quite where they were going to start ramping it up again for the next season. And then after a couple of years, H1N1 vaccines started to become part of the mix of how influenza vaccines work. And yet, even then, even when the entire influenza vaccine infrastructure, which is the largest, the most robust vaccine production infrastructure on the planet, even when that was restructured just for H1N1, there was a lot of consternation and maneuvering because of supply issues to get H1N1 vaccine on site, and a lot of broken contracts and hurt feelings internationally when that happened. So you're right, it's hard. Merck, who's been very committed to their recombinant vesicular stomatitis virus vaccine for Ebola, they actually ended up, in order to get that fielded, they had to go looking for facilities that could be purchased and repurposed in order to produce the vaccine safely. And that safety aspect is a big one. Every time you change any small aspect of the vaccine, whether it's the material that's creating the immune response, the material it sits in order to get into the body best or be stored, the aspects of it that help irritate the immune system so that it's ready to look at what you're giving it, the protein, the adjuvant. Anytime you change any of that, you have to completely revalidate the safety of the system. And we've learned that the hard way over many, many years that has to be done. But it does mean that production is always a concern and a consideration, which is one of the reasons why going after multiple ways to develop multiple types of vaccine for a threat that you care about and you think is going to be at scale, need lots and lots of product. It's one of the reasons to advance several ideas. One of those reasons is just because ultimately many of those ideas fall away because they were great, but you couldn't execute them yet. Yeah. Either the lack of technology or just because you weren't able to observe the effect that you needed. But the other reason is manufacturing. Many of these places are a cell-based vaccine production capability is very, very different than some of these other technologies, for instance, that do virus-like particles. So that helps too in mitigating it.
Well, and we have 3 minutes left. So we talk, developing, approving, hoping that there will be enough to go around. But then you have that whole idea of the delivery system too. So again, less than 3 minutes remaining. Talk about the importance. I mean, it's great to have all the others, but unless you have a way to effectively and efficiently get it to people, we're almost talking like a moot point here. So tell us about the delivery.
Well, we're very fortunate in the U.S. that we deliver a lot of vaccines all the time, and we do it both through public and private mechanisms and pharmacies, and we have a lot of experience in communities getting vaccine to people. And so I think we're really blessed, actually, in that capability. It's also true that whether it has to be very, very cold, whether it has to be mixed on site with a solution makes a difference. The cold chain issue in the U.S., we're very fortunate with that. Globally, that's a other thing entirely. And getting to people in communities at risk, figuring out ways that it can be stored so you can surge production are all challenges. So you're absolutely right. Supply chain and delivery matters. And it's another reason to invest regularly in health systems and public health capabilities that allow the delivery of what you need every day that can be adapted for what you need in emergencies.
Okay, so Dr. Brett Major, with our viewers watching across the state, I just want to kind of bring this to a close. Talk to them for a minute. Of course, everybody's coming from different places. Final thoughts on this race to a vaccine for our viewers and everyone else.
Well, hopefully we're going to have vaccine candidates here in the next year, and we'll see them start to roll out. But regardless, even in the setting of a vaccine, it is respecting yourself, your neighbor, and the virus, hand washing and distancing, being mindful when you're ill, reporting early to care, thinking about your health and your needs and your neighbors, and being respectful in ways that remember that just like seasonal influenza, and certainly now with coronavirus in this emergency, it matters to be thoughtful.
Okay, thank you. We have a couple of graphics that we want to put up for our viewers in the event that they want more information. We are, of course, talking about the race to a vaccine today. The first graphic is Centers for Disease Control and Prevention. That number is 800-232-4636, and that website is cdc.gov coronavirus. The second is Nebraska Medicine. That website is nebraskamed.com/patients/COVID-19. Dr. Brett Major, thank you so much for joining us today. We want to wish you and everybody else who's working on this the very best, and thank you again for sharing your time and expertise.
Pleasure. Thank you.
All right. And I want to thank you all for watching. We'll see you next week.
Consider This is an issues and information program produced by UNO Television. More information and program archives can be found at netnebraska.org/Consider This. If you'd like to share your comments about this program, call UNO Television at 402-554-2516 during regular business hours, or send an email to cwyatt@unomaha.edu.